Hello and welcome to our poster presentation on the role of free-floating macrophytes in sequestration of E. coli in the Scannick River in Connecticut. My name is Kirsten Martin and I'm Associate Professor of Biology at the University of St. Joseph in Connecticut, and this project was completed with help from a high school research student, Lucy Noble. The Scannick River has been monitored for a long time for water quality, close to 20 years. It's been monitored monthly for nutrient levels. And then beginning in the summer of 2015, several sites were selected to have additional weekly monitoring during the summer months for E. coli levels. The lower section of the Scannick River is listed as an impaired waterway by the Connecticut Department of Energy and Environmental Protection, and it's listed as impaired due to high levels of bacteria, although high levels have been found in other sections of the river as well. One location, the Summersville Mill Pond, is a very unusual location in that it consistently has low levels of E. coli, despite elevated levels of E. coli being found immediately upriver and downriver of the location. One thing that's unique about this particular site on the river is the presence of extensive free-floating vegetation. And since there has been numerous research studies looking at the ability of terrestrial systems and vegetation in terrestrial systems to sequester E. coli, we were interested in looking at the ability of aquatic vegetation, particularly free-floating vegetation, to do the same thing in aquatic systems. So this study was taken on during the summer of 2018. And during the study time, weekly samples of both water and aquatic vegetation were collected and then processed to determine levels of E. coli present. Give you a little bit of background about the Scannick River. The Scannick River watershed itself drains 90,000 acres in Connecticut and neighboring Massachusetts, and the river is approximately 38 miles in length. There are several small tributaries that are classified as Class A water quality, which is water suitable for drinking, although there are several locations on the main stem that have significantly impaired water quality. The Scannick River's headwaters are in Stafford, Connecticut, then it heads north into Hamden, Massachusetts, before returning to Connecticut and flowing through the towns of Somers, Enfield, East Windsor, Ellington, and South Windsor, where it joins the Connecticut River. Just a note that in this research project, we focused on E. coli in general, although there are specific strains of E. coli that are pathogenic and cause a significant human and animal health risk. We are just focusing on overall E. coli. So this is an image of our sampling location, the Summersville Mill Pond. As you can see, this is where the main stem of the Scannick River flows into the Mill Pond impoundment area. Then we have the dam at the far end, and then the rest of the Scannick River is downriver. You can see from the image that there are extensive vegetation mats, some of which have collected sediment and have formed islands, but most of these in this area are the larger free-floating macrophytes that we were interested in looking at. So during the collection period, we used a dip sampler to collect the water as well as the vegetation. They were collected in the same sampler, and the sampler was sterilized between collections to try to avoid contamination, and gloves were also worn. We also collected information on water temperature, air temperature, and pH at the time of collection, and then all samples were delivered to the University of St. Joseph laboratory for further processing. In the lab, we did need to do some pre-processing of the vegetation prior to putting it into the colert, because we were interested in only looking at the bacteria that was within the plant tissues and not on the surface of the plant. So we initially rinsed off any attached material, any attached soil with the eye water. Then we did a flash sterilization using a 5% bleach solution. We allowed the vegetation to sit in that solution for 2 minutes. And then we re-rinsed the vegetation in DI water to remove any of that bleach solution. 5 grams of the sample was then measured out and placed into a sterilized blender so that we could disrupt the cell walls. Then we added a packet of pre-measured color reagent to 100 milliliters of the sample, poured that 100 milliliters with the reagent into a quanta tray. This is what the quanta trays look like. And then incubated that tray at 35 degrees Celsius for 24 hours. Following incubation, we place the tray under a UV viewing cabinet and count the number of large squares and small squares that are fluorescing. And you can see in this image what those fluorescing squares cells look like. The water sample did not need any pre-processing, so we just decanted 100 mils, added the same type of pre-measured colorant reagent, put it in the tray, and then also looked at number of fluorescing large and square cells. And then the number of large and small cells were looked at compared to an MPN table, most probable number table, and that's how we determined the number of colony forming units of E. coli in both vegetation and water. So to get to our results, to make it a little bit easier to see some of our results, I have color-coded the results. So all of the water results are in blue, and then all the vegetational results are in green. So as you can see, there is a lot of variation from each collection session. So samples of water and vegetation collected from the same date are adjacent to each other. So there is a lot of variation during our study session. We did do some statistical analysis of the data, and it did not result in any statistically significant differences, but there's still some interesting trends and interesting findings to find from our results. We do see a lot of variation, as I mentioned, in both the water and the vegetational amounts of E. coli, and that's to be expected particularly for the water. This is surface water. It is open to any kind of contamination. Rain events, as you can see from our table, following rain events, we did get higher levels of E. coli flowing into the system, so we expected that variation in the water. But what is really interesting is to see as great of a variation present in our vegetational samples, because again, this is E. coli that was internal to the cell. This is not surface E. coli. This is what was being held within the tissues. So there is some suggestion that the vegetation is responding to changes in the environment, whether it's needing to bring in more E. coli, and E. coli is serving some kind of a beneficial role to the plants, we don't know. But we would have expected to be very little variation in the plants if the plants were not responding to the environment. It does seem that they are responding in some way. So in conclusion, while none of the associations that were recorded in the study are statistically significant, that does not mean they are without value. There's definitely some interesting trends that need to have further study, looking at the ability of various types of free-floating macrophytes to sequester E. coli. And also there needs to be further research in looking at the maximum amount, how long these plants can potentially hold on to E. coli, as well as other nutrients or pharmaceuticals that might be in the water system. Further research was conducted by a different student looking at attached vegetation, so rooted vegetation versus free-floating versus riparian, and there were some very interesting and statistically significant associations between different types of plants and levels of E. coli. And another project was conducted just looking at riparian zones and the types of vegetation, annual, perennial, trees, shrubs, lawn grass, and the difference in abilities of sequestering E. coli between those types of vegetation. Again, very, very interesting results that were that showed some statistically significant impacts. So with that, I'd like to thank you for listening to our poster presentation, and feel free to send me any questions. I've included my email contact on this slide. Feel free to send me any questions you might have about our research. Thank you.